Hello and welcome to my latest blog. Today we're going to talk about the plain language summary of my latest paper, Mapping the Phylogeny and Lineage History of Geographically Distinct BCG Vaccine Strains. So this is a plain language summary of a paper that so myself and colleagues from the Centre for Clinical Microbiology at UCL in London, wrote. And the paper looked at a collection of different versions of the BCG vaccine that we have. If you want a link to the original paper, you can find it in the blog. It's open access, so anyone can read it. Firstly, a little bit of a background to the study. The BCG vaccine, or Bacillus calmette-guerin, has been used for over a century to try and prevent tuberculosis. BCG is an attenuated or weakened form of Mycobacterium bovis, the bacteria that causes cow tuberculosis, and is genetically very similar to human tuberculosis or Mycobacterium tuberculosis. You can learn more about the disease tuberculosis in the link in my blog. French scientists at the Institut Pasteur grew Mycobacterium bovis for many, many years, 13 to be precise. These bacteria take a really long time to grow, and eventually it mutated enough for it to no longer cause disease. It was trialled in animals and then humans before being rolled out as a vaccine. And when the success of the vaccine became apparent, batches of the live bacteria were sent out to different laboratories around the world, Japan, Denmark, and Canada, to name but a few, so that they could all grow their own vaccine and protect their own populations from TB. And in the 1900s, TB was extremely common all over the world. Because these different laboratories used different culturing methods to grow BCG in the early 20th century, the bacterial strains from different geographical areas have become genetically different. If you want to learn more about the history of the BCG vaccine, you can read about it in the link on my blog. How well the BCG vaccine works to protect people from tuberculosis seems to vary around the world, and this is thought to be due to a number of different factors, including the other infections that people might commonly be exposed to, such as malaria, parasites, or other mycobacterial species, the climate, general immunity, and whether people live in densely populated, poorly resourced urban settings. It's also thought that the genetic and phenotypic, so phenotypic is the physical changes, like the shape of the bacteria when they grow, these variations in BCG, so the bacteria itself, caused by years of growing different strains in different ways, may also play a part in how well BCG works as a vaccine, as different countries use different strains in their vaccination programs. You can see who uses what on the very interesting BCG World Atlas website, and the link is in the blog. The geographical strains can be roughly categorized as early, or those taken from the original Pasteur strain before the mid-1920s, and late, those taken afterwards. Early strains are thought to be better at providing protection from tuberculosis, but also cause more side effects because they are genetically more similar to the disease-causing M. bovis from which BCG originated. In the 1960s, the World Health Organization realized that these differences in the BCG vaccine strains needed to be addressed. You can read more about the standardization of BCG vaccines here in the link on my blog. The WHO committee decided that countries weren't allowed to just keep growing vaccine strains with their own methods indefinitely anymore, and that they had to freeze down stocks of it and then take cultures from that stock. Once growing in media, laboratories are not allowed to subculture or move into fresh media more than 4 times, and this stops the bacteria from developing genetic mutations, which is a common and completely natural occurrence and the basis how life evolved. The development of genetic sequencing in the 1970s began a new era of understanding living organisms, and soon the commonly used, commercially available BCG strains have been genetically characterized. As a result of the WHO's standardization research, a colleague of mine at the Centre for Clinical Microbiology, Dr. Helen Donoghue, acquired a collection of different BCG strains well over 50 years ago and has kept it ever since, for a while in her basement and then in the cold room in our department. What's really interesting about her collection is that it's not just the commonly found commercially available reference strains of BCG, such as BCG Danish 1331 and BCG Japan 172. The numbers represent how many passages or subcultures the strain went through before it was frozen down. There are some weird and wonderful research strains that may have never made it into the arms of the population in our collection. And whilst the common ones have been sequenced, the more unusual ones don't appear to have been yet. If there are a lot of different if there are a lot of differences in these strains, especially in certain genes that might make the strain more effective as a vaccine, it might be of interest to other tuberculosis scientists. In fact, in over 100 years, nobody has come up with a better vaccine for tuberculosis, which means any new piece of information about BCG could really be useful. Aim of our study. Whilst we've had this collection of BCG strains for half a century, it's only in recent years, with the introduction of next generation sequencing, that it's been cheap enough to sequence our isolates. So we aim to see if we could grow the bacteria directly from the powdered cultures in the vaccine vials, extract the DNA from them, and sequence them using Oxford Nanopore's MinION device. Methods. First, we had to sort through our big box of vaccine vials and catalogue what we actually had. Once we worked out how many vials we had for each different strain, we took one of those vials and cracked it open, and it felt a little bit like opening a time capsule. We then added phosphate buffered saline, or PBS, which, unlike water, doesn't pop cells due to osmosis. We let the PBS soak into the dried bacteria for a few minutes, and then we put some of that into a liquid culture, some of it onto an agar solid culture, and some we saved to extract the DNA from in case we couldn't get them to grow. The extracted DNA was quantified, and we did whole genome sequencing on as many as we could. We then analysed the sequencing data that we got by looking for mutations, specifically in genes of interest, such as those related to virulence, the ability of bacteria to invade host cells, growth, and cell wall structure. We also compared the different strains to see which were more closely related by aligning them using a reference genome. The best known and genetically understood BCG strain, and therefore the most commonly used as a reference, is BCG Pasteur. Now, this is not the original one, sadly, which was lost in a fire many years ago. It's the newer version. We also looked at regions of difference, which encode antigenic. So, antigenic is something that induces a response from the host's immune system, antigenic proteins that could be promising vaccine targets. Results. We found that we had 50 geographically distinct strains in our collection, and out of those, we managed to revive and successfully culture 36 of them. And I was genuinely pleased about this. The sad bacteria had been stored as a dry dust since the 1960s, and it seemed a genuine miracle that anything might grow. We managed to sequence 38 of them, so we couldn't get a couple of them to grow, but we did manage to extract DNA directly from those vials successfully. We then looked at the strains that did grow and found they look physically different. Those taken from the original Pasteur strain before 1934 had a flaky appearance that looked almost like dandruff in a liquid culture, and those taken after 1934 had a more clumpy breadcrumb-like appearance. And you can see photos of the flakes and clumps in the supplementary material at the end of the paper. When we put all of our strains into a phylogenetic tree and compared them with other BCG strains that had already been sequenced, the strains from different geographical areas tended to cluster together, most notably BCG Russia, BCG Japan, and BCG Danish. This makes sense because they will have been grown in the same labs under the same conditions, and might possibly even been from the same culture tubes. When we looked at mutations and parts of the genome that were missing, we found that the late strains had lost regions of difference 2, 171, and 713. We found that the early strains had mutations in genes SIGK, PLA-A, MMA-A3, and ECCC5. So the fact that we had mutations in early strains is because the reference genome was BCG Pasteur, which is a late strain. So the later strain ones will be more similar, and the early strains will be more different. PCAA and MMA-A3 are important for cell wall growth, and there has been other research showing that the late and early strains have differences, which could be important for cell growth and the bacteria's ability to invade host cells. SIGK is a positive regulator for the antigenic proteins MPT70 and MPT83. And mutations in SIGK in M. tuberculosis show variable production of these proteins. In M. tuberculosis, MPT83 which is a surface lipoprotein, has been shown to be one of the strongest Th1 cell antigens, which means that the Th1 response is strongest to these proteins on the surface of enteroberculosis. So, if a bacteria has the early version of the SIGK gene, this might play a role in the greater immune response thought to be produced by the early BCG strains. We also found two different variations of the gene ECCC5, this is a mouthful, in these early strains. This gene hasn't been as well described in BCG, but it does appear to encode a protein in the ESX5 membrane complex secretion system. Secretion systems are needle-like structures that are formed on the surface of many bacterial species that allow them to inject proteins into host cells. So these proteins, called virulence factors, help the bacteria take over the cell. You can read more about bacterial secretion systems in the link in the blog. The ESX5 secretion system is fundamental to mycobacteria host cell interactions, and deletion of the ESX5 system from M. tuberculosis is being tested as a vaccine. Conclusion. Whilst we found that BCG Russia, BCG Japan, and BCG Danish showed strong clustering on our phylogenetic tree, the late strains, including BCG Pasteur, showed a bit more variation. We found a wide range of SNPs, so single nucleotide polymorphisms or mutations, in the geographical strains. So we found as much variation between the different strains as within the different strains. And there were lots of variations or lots of differences within BCG Danish, as well as lots of differences between Danish and Russian. The mutations that we found suggest that there may be some interesting differences in the host immune response when infected with the different strains. In fact, we're about to start testing this out on some macrophage cells, and these are the immune cells that tuberculosis or M. tuberculosis and BCG invade when they enter the human body. So we want to do that to see if there are different responses. So watch this space.